You've probably heard the human genome called the Book of Life, right? A massive text with 3 billion letters. But what if it's not just a book? What if it's more like a piece of sophisticated code, governed by a hidden mathematical language we're only just beginning to decipher?
So that's the big question, isn't it? The one we're really digging into today. Are we looking at a beautiful, chaotic jumble of genetic info that just happens to work? Or is there a deep, almost impossibly elegant structure controlling the whole thing from the top down? Okay, so before we dive in, let's get on the same page with the basics. The language of DNA isn't written in letters, but in 3-letter words called codons. The alphabet only has 4 letters: T, C, A, and G, which gives us 64 possible 3-letter words. These codons are the absolute foundation of our genetic code.
All right, so a language in our DNA? If there really is one, we're not going to find it by staring at individual letters. No, we have to zoom out and look for patterns in how these 3-letter words, the codons, are actually used across the entire genome.
This really sets up the big debate. On one hand, you have the idea that the genome is just the result of eons of random mutations that somehow, added up to us. But on the other hand, there's this wild idea that it's a finely tuned system where every single word follows a precise mathematical blueprint. Now, to be fair, we've known for a long time that the genome isn't completely random. Take Chargaff's rules. They show us that the amount of the letter A is always pretty much equal to T, and C is pretty much equal to G. It's our first big clue that there's some kind of order at play.
But that's just letters. What happens if we zoom out from single letters and start counting the words, the codons themselves? Well, this is where the story gets really, really interesting. So, back in 2010, a researcher named Jean-Claude Perez did something that sounds simple, but was massive in scale. He took the whole human genome sequence, all 3 billion letters, and just counted. He tallied up how many times each of those 64 codons appeared.
And wow, this is what he found. The result was anything but random. If it were random, you'd expect a smooth curve, right? But that is not what we see. Instead, the codons clump together into these distinct pairs. See how the black and gray bars are almost always the same height? It's an unbelievably orderly pattern that just begs for an explanation. Let's just look at the raw numbers here, because this is wild. The most common codon, TTT, shows up over 109 million times. And its partner, AAA, almost the exact same. And it just keeps going. ATT and AAT, TCT and AGA, the counts are so close it's almost spooky. The big question is, why? What's the connection between these pairs? Well, to crack this code, the research takes a turn into a place you would probably never expect. A beautiful and seemingly totally unrelated branch of math. We're talking fractals. Specifically, something called the dragon curve. I know, this looks super complex, but the idea behind it is actually something you can do right now. Just imagine taking a strip of paper and folding it in half, then fold it in half again, and again, and again. When you finally unfold it, the pattern of creases you've made is this fractal. It's a dead simple process that creates mind-bending complexity. Okay, so how on earth do you apply a paper folding pattern to DNA? Well, the method is actually pretty genius. Step 1, you lay out all 64 codons in their standard grid. Step 2, you use the folds of that dragon curve to split the grid in half, then split those halves in half, and so on, 6 times over. And this is basically what it looks like. Each fold of the fractal slices that codon grid into 2 groups, which the paper calls odd and even faces, the dark and light sections you see here. This gives us 6 totally different ways to divide the 64 codons into 2 teams of 32. But the real question is this just a fancy way of drawing lines, or does it actually mean something? Oh, it means something. It means everything. Because when you add up the total counts of all the codons in these groups and compare them, you find something incredible, a stunning mathematical constant that's been hiding there in plain sight the entire time. Yup, that number, the golden ratio, phi. This is one of the most famous numbers in all of mathematics. It shows up in spiral galaxies, in seashells, in classic works of art, and now it looks like it's woven deep into the fabric of our own DNA. So here's the absolute game changer.
When you calculate the ratios between the even and odd codon groups for each of those 6 fractal splits, the numbers aren't all over the place. They lock on to 2 very specific values, which the paper calls attractors. It's like the whole system is constantly aiming for these 2 precise mathematical targets. I mean, just look at the data for yourself. The first fractal split gives a ratio of almost exactly 1. The second one gives a ratio of about 0.691. The third, back to 1. The fourth, back to 0.691. You see that? It's a perfect alternating pattern. You just can't make this stuff up. This is undeniable mathematical order. And here it is. This is the punchline. The first target number is just 1. Perfect balance. But the second one, that 0.691 number, it's a precise formula derived directly from phi, the golden ratio. It's not close. It's exact. The entire structure of how our genome uses words is being tuned by these 2 fundamental mathematical constants. Okay, deep breaths. We found this unbelievable mathematical pattern. But so what? What does it actually do? I mean, a pattern this precise has to have a purpose, right? It can't just be a pretty coincidence. Well, the paper proposes a grand theory for exactly that. And to get there, we have to circle back to the very beginning. Remember those pairs of codons with almost identical counts? Well, it turns out they're what's known as mirror codons. And a mirror codon is just what you get when you reverse a codon's letters and then swap them with their opposites. It's a really simple 3-step recipe. Let's take the codon TCG. First, you just reverse it, so you get GCT. Then you apply the base pairing rule. C swaps with G, and T swaps with A. So GCT becomes CGA. That means the mirror of TCG is CGA. And what the data shows is that for basically every single codon, its population is almost identical to its mirror. So, putting it all together, here's the theory. The genetic code table isn't just a simple dictionary for translating genes into proteins. It's also acting as a global checksum matrix. Now, if you're into computers, you know a checksum is a simple way to check for errors. This theory suggests our genome has a built-in, mathematically perfect system for maintaining its own structural integrity. Perez uses this perfect analogy to explain it. He says the genome is like a deck of cards, right? When you deal the cards out, the sequence looks completely random. But you know that the deck as a whole has a fixed, perfect structure, 4 aces, 4 kings, and so on. In the same way, the sequence of our DNA might look chaotic, but its total makeup obeys this incredibly precise, hidden order. And that just leaves us with this final, totally mind-bending thought. The golden ratio is everywhere in nature, in patterns of growth, of beauty, of the cosmos. So what does it mean if the very code of life itself is fundamentally tuned to that exact same mathematical harmony? What other secrets are still hiding in the book of life, just waiting for us to figure out how to read them?